Build my home on hollow ground. Too proud to grow.
This is Craig Smith with a podcast about artificial intelligence. This week I entered the smelliverse, the dimension of odors, with Alexei Koulakov, a professor at Cold Spring Harbor Laboratory on the North Shore of Long Island. Alexei uses machine learning and other tools to explore the least understood of our senses, the sense of smell. I've talked to people on the podcast about computer vision, computer hearing, and even the sense of touch in robots, but I'd never heard anyone talk about using machine learning to explore the sense of smell until I met Alexei. He's working to unlock the mechanism by which we perceive odors and classify millions of volatile molecules by their smell. It's a fascinating subject, and will have you sniffing the air at odd moments, increasing your awareness of this mysterious dimension. I hope you enjoy listening to Alexei as much as I did.
Cross it to find your synthetic light.
So I thought I would get you to sit down and talk about the physics of odor or the biology of odor perception and how you've been able to apply machine learning to classify odors and that sort of thing. But to begin, can you sort of tell me about yourself, how you got into this, what's your educational background?
I was trained as applied mathematician, engineer, and then I became theoretical physicist when I moved to the States. And then throughout my life, I had very strong interest in biology, right? And particularly in how the brain works, neuroscience. And for some reason, I got lured into neuroscience from theoretical physics. Although I did actually hold a faculty position in physics, but I started doing biology. But then I decided that it's better for me, basically to switch to biology, to neuroscience completely. So I went to Cold Spring Harbor to do it, But I do something that is called theoretical neuroscience.
Called
Theoretical.
Theoretical
Theoretical / computational neuroscience, which means that I'm trying to understand what are the theoretical models that can explain brain function. So that's the overall idea, and I basically apply methods, various methods that I learn in, computer science, theoretical physics, math, applied math, to understanding the brain function. That's what I do on an everyday basis. It's the best job in the world, right? And I think the most exciting part of that job is that you get to learn a lot because it's a very diverse field, and that's basically the thing that drives me.
And then out of that came an interest in
olfaction. So olfaction basically is the least understood of our senses. One would say that vision is poorly understood, but the high level vision is poorly understood. On the low level, we know how photoreceptors in the eye, how they interact with light, right? But in olfaction, we don't have even that basic understanding. So we don't really know the mechanisms by which molecules activate olfactory receptors. I mean, olfactory receptors are interesting because they're very similar to the ones to photoreceptors in the eye, so they're related. There are much more olfactory receptors than we have three-color vision, right? So we have three types of photoreceptors in the eye. And we have about 350 of olfactory receptors. So roughly speaking, olfactory system is a retina with 350 types of colors.
Wow.
And maybe 4 pixels. And it's not clear, in the retina we have about 1 million pixels.
Right.
In olfactory system we have about 4 pixels, so the spatial resolution is really limited. But most of the resolution in olfactory system goes into this color in quotation science that basically allows us to recognize different molecules.
Yeah.
But what is unclear is what type of molecules, what type of features in the molecules olfactory system is sensitive to.
Why would evolution develop kind of an imbalance between what you're calling the pixels and
the spatial resolution versus the feature.
That's right.
Feature space versus the spatial. I mean, this is not really clear. I think overall the spatially olfactory stimuli, which are streams of odors in the environment. They're not so precise, they're very dispersed. But again, I'm speculating here. Like there are a lot of interesting interactions between, anecdotal kind of interactions between the visual system and olfactory system. Because, like, for example, if you take a mouse, it would have about 1,000 olfactory receptors. And humans have about 350. So we lost a lot of olfactory receptors in the course of evolution. And so there is a suggestion, which is backed up by very strong evidence, that we lost those receptors due to acquisition of color vision. So we don't need them anymore because whatever the function that they played is taken over by the color vision that we have, by trichromatic vision. Like, for example, we have monkeys. There are monkeys, New World monkeys that live in Americas. They have dichromatic vision, and they have a larger number of olfactory receptors.
Wow, that's fascinating.
So there's a weird interaction between visual and olfactory system.
Yeah.
So another interesting interaction, again anecdotal, is that, I mean, the receptors actually in the eye and the receptors that olfactory system is using, I mean, they're proteins which are on the surface of the cells, right? And they interact with various, stimuli. So they're very similar genetically. They both belong to the same type of receptor. And interestingly, the visual, that olfactory receptors, they bind odorant molecules. And, by the strength of the binding, by the affinity of this binding, they recognize what the molecule is. That's basically how it works. So some receptors don't bind certain types of molecules, and that information, or some other receptors will bind the molecules, and that information basically is used to derive what is the stimulus, right? Which receptors are activated, which are not. So the same thing happens in visual system, because visual system, protein in the visual system actually relies on small molecule which is bound to that protein, which is derived from vitamin A. So that small molecule, so essentially, like, the visual system protein is an olfactory protein that binds another small molecule called retinol that changes conformation when the light is coming in at certain wavelength. So, and that's how we recognize different colors.
That's interesting. The number of receptors in the human olfactory system, you were saying in a mouse it's
about 1,100.
Right.
So it's about 3 times more.
Right. So does that mean that the mouse can differentiate between many more molecules?
Oh, yeah.
Yeah. And I've heard dogs as well.
Dogs actually have slightly fewer receptors, surprisingly, than the mouse.
Oh.
So it's surprising that dogs are, actually hunting mice rather than the other way around.
Yeah.
But dogs have much larger olfactory epithelium, meaning that they have more cells basically in their way. So probably the threshold for just detecting odorants is lower. So they're more sensitive in that way.
Yeah.
And again, I mean, those systems are very strongly fine-tuned because those two animals, those two species, their existence depend on the sense of smell, right?
Yeah.
And when I say that it's the most mysterious of senses, I'm being honest. We don't really understand what are the evolutionary pressures.
Yeah.
That's what we're trying to understand, actually, using machine learning and all other approaches.
So the machine learning application that you've worked on, what kind of sensors are you using to detect the molecules?
That's the thing. So there's no technology hasn't developed sensors which parallel in their sensitivity and discrimination the real biological sensors. So we're not using any sensors. So what we actually try to do is we try to predict human perceptions of various molecules, and we're trying to understand what are like the evolutionary mechanisms or the evolutionary pressures on those 350 receptors that we have, right? So, and can we describe those pressures using machine learning language? So we don't use the measurements of which are obtained from sensors, because the sensors they're basically indiscriminate. The sensors that we have, the best ones, are not really good at discriminating odors. So the type of data that we have, there are 2 types of data. There is data conformations of like basically shapes of molecules. Mm-hmm. And I mean, you might wonder how is it useful, right? Because it doesn't have any olfactory perceptions. You just have like molecules and how they exist, how they are shaped in 3D space. But it turns out that for machine learning, this is actually very useful information. And I'll describe later how, right? But that's one of the types of data that we have. And this is virtually unlimited data set because we know everything about all the molecules. Like chemists are not wasting their time. Yeah. They're computing. So we have about 10 million molecules in our database that we know everything about. We know conformations, we know their physical and chemical properties. So it's an enormous data set. Unfortunately, we don't know how they smell. So we know how molecules smell for about 4,000 molecules, right? But let me actually go back. So what we're trying to do here and what is really missing, the real question is the analog of the color space. Like in the visual system, we know that there are red, green, and blue, basically, right? And we can synthesize stimuli based on that knowledge using like a TV screen, right? So the fact that we have such an efficient synthesis of visual stimuli is a testament to our knowledge, to our understanding of the color space. Yeah. That it's basically 3 colors, right? And we don't have a similar understanding in olfactory system. So we don't really know what are the features of odorants that olfactory system recognizes, and if there is any simplifying structure in that enormous space of molecules. I mean, there are about 10 million volatile molecules. Is there anything that we can, like, do we simplify it, or we just like basically recognize every molecule as a new, unique, entity? So that's the basic question. And so there is a hypothesis that we're trying to formulate that there is a simplifying structure. There is something like red, green, and blue in olfaction, and basically try to understand whether what we say olfactory perceptual space is low dimensional, right? Like the color perceptual space is 3-dimensional. So the question is if it's possible to describe the space of percepts of molecules, human percepts of molecules, by a low dimensional, by a small number of parameters, right? So that's the hypothesis of low dimensionality of olfaction. And it's interesting that to some degree you can like take the like olfactory and perceptual data set and try to reduce dimensionality, and a lot of information can be captured by low dimensional projections of those percepts. And the interesting thing is, what are those dimensions, right? So like what are the analogs of red, green, and blue? And we don't really know, and to this point, like how to attach an anecdotal meaning to them. Although they do exist in computer, we don't really understand what the meaning of them, with a small number of exceptions. So one of the most interesting dimensions in olfaction turns out to be pleasantness of a molecule. Pleasantness, how pleasant the molecule is. This is something that, like, for many molecules, people find universal. Like many molecules are uniquely pleasant or unpleasant, That's like basically one of the most dominating dimensions. But perhaps if I were to bet, I would say that there are probably about 10 dimensions, 10 analogs of red, green, and blue. And if that were the case, if that was the hypothesis that would be confirmed by data, that would be a big deal, I think, because that would really drive the research, trying to understand what those dimensions are. Another question you can ask is, how do you describe olfactory percepts, right? So this is something that exists in our head. Right. How do you put it out there? How do you put it in the data set? Like when you smell a rose, how do you how you convert that into data, right? This is actually a very imperfect process at this point. There are many different methods. Like, for example, people could try to compare two molecules, how similar they are, right? And then probably if they're similar, more similar, then you will find that they're closer in this perceptual space. I have a term for this perceptual space. I call it smelliverse. Just basically an olfactory color space. Right. You could try to find similarity between percepts for pairs of molecules, and then to build this smelliverse from inside, just based on that distance information. And that, surprisingly, that strategy fails dramatically, because no people will ever agree on how
Right.
To compare apples and like smells of apples and oranges. So if you do that, then the only thing that people can agree is how pleasant things are. Turns out, what you will see is the smelliverse is one-dimensional. It will be basically the pleasantness will be the only dimension, and the other dimensions will kind of average out. They will be washed out by this lack of consensus, if it makes sense.
Yeah.
So that turned out to be not a good strategy unless you are measuring like a pleasantness of molecules. If you are like specifically interested in how pleasant the molecules are, that would be a bad strategy. So another method is so-called profiling, and that probably generates the best data sets right now. And that is describing molecules with words. So there are carefully selected hundreds of words, and then you basically ask humans to say which word applies to which molecule.
One word per molecule, or which words?
Which words? I mean, normally, like in the data sets that we use, there are like on average 2 to 3 words per molecule, and there are hundreds of words, like literally.
Right.
And those data sets are built by olfactory experts, so they kind of know where those words are located and how to describe molecules. So that is very imperfect type of data because it's semantic.
Yeah.
Right. It relies on the ability of people to describe the indescribable percept that exists in their heads with limited capacity of, semantic capacity. So, and then the third type of data, which is now appearing, is actually imaging from olfactory cortices of mice. So that would be actually a better descriptor, but we don't have much
Imaging
data.
of...
Just imaging of like a mice would sniff a molecule, and then you would basically install a microscope in the piriform cortex, which is olfactory cortex in the mouse, and you would image the activation pattern of cells in the piriform cortex. And that is basically, I think it's the ultimate measurement. So it's not clear how it's going to fail at this point. The way it fails at this point is that we don't have much data for many molecules so that we can train machine learning models.
You could have analogous data from human cortex scans, no?
No. That's a very good question. I mean, you're asking a very good question. But the idea is that why is it not possible comes from the way how cortices are activated by smells. It turns out that essentially when you present the smell, cortex will respond in a white noise manner. So it will look like every small area is activated, it contains activated cells, and they're scattered around like pseudo-randomly. The pattern is reproducible, meaning that if you present the same or like a similar smell, a similar set of patterns, a similar set of cells will be activated. Overlapping set of cells will be activated. But it's salt and peppery, so you basically see like a salt that's scattered all over the piriform cortex, right? So you need to have a very high spatial resolution to study that pattern. And like if you try to use methods that are available in humans, such as fMRI, they have a larger magnetic resonance imaging. So you will see the spatial resolution is not enough really to resolve the subtleties of that pattern.
But in a mouse...
In the mouse, you can install a microscope near the cortex and see a small chunk of that activation pattern, but at high spatial resolution.
Literally a microscope.
Literally a microscope, yes.
And how does the activation visualize then?
There is a fluorescent dye that is activated by calcium. So when the cell responds,
I see.
There are some like calcium is released from the internal stores of the cells, and that release of calcium activates a fluorescent reporter that fluoresces, and you can detect that. It's a specially designed mouse. It's not a regular mouse.
Yeah, that's fascinating. And so what do you do with this data set? And you don't have enough data yet.
That data set, although available, it's not really useful at this point because they're literally like data for maybe like 20 smells, and like machine learning models, they rely on thousands of data points to be useful or predictive. So I think this is like the future, but at this point we don't have that data. We have data for humans.
Yeah, and that's the semantic data.
Yes.
Yeah, and you average that, and you could come up with some sort of a it's not a high resolution, but you do come up.
Well, it's not actually so bad. I mean, it's not so bad because it's human, right? So that's what we're interested in. We're not really interested in mouse olfaction, right? Although that's interesting on its own, right?
Yeah.
And like another thing which is interesting about olfactory system is that it's very similar between like vertebrates, invertebrates. It goes back to basically to some very low level organisms such as flies. Like flies, for example, fruit flies, they have a very similar olfactory system. They have very dissimilar visual system, but the anatomy, the structure of olfactory system are basically similar to humans, so which is fascinating. So once you understand one, so there is basically this notion that if you understand one, you can generalize this knowledge to another, you know.
Using the semantic data that you've collected, how large is that data set?
So we didn't collect it. We used the efforts of, olfactory researchers. I mean, we're computational people. We don't really collect data.
Yeah.
So it's a very good question. There are several data sets, and they're different quality. So I think the best data set that is available right now is collected by a company, which is called GoodSense. GoodSense. It's freely available for downloading. It's basically about 4,000 molecules, a little bit less than 4,000 molecules. They are projected to about 600 words space. So, and again, it's discrete. It's mostly like zeros with a few ones corresponding to the words that describe that molecule. So it's basically a huge lookup table. Like for every molecule, some number of words varying from 1 to maybe 8 that describe that molecule.
But if you're training neural net, I presume you're using deep learning on that data set, then what are the inputs because you're working toward a classification system.
Right.
So what are the inputs then? What are the classifications? Are you classifying specific molecules or families of molecules?
Yeah, we actually do two things. We are very good at one thing, and we're not so good, we're not perfect at another thing. And I'll describe you what those things are. So how does deep learning apply to this? Where does it come in, right? So the main hypothesis that we have is that olfactory receptor is basically, this protein is basically a spatial filter that interacts with molecules and extracts features from the molecules, certain chemical features, right? So it's a spatial filter, and you know that like in the visual domain, the deep learning networks, they are very successful in classifying images, right? So our idea is basically this protein is a network. Is a network of domains, the network of different parts that interact with each other. I guess network is another way of describing interactions, right? So the olfactory receptor is a network that describes the filter that interacts with molecules. It extracts certain molecular features. And that's how we can use deep learning. That is our hypothesis. So that's the overarching hypothesis, and we use it in two different applications. And one application is to describe the space of molecules, of all molecules in the universe which are volatile. And how would you do it? And to do that, we do something which is called autoencoder. So the idea is that you present olfactory receptor as a neural network, very similar to the visual network, except for it's three-dimensional. And so you present this molecule to the network, and what it does, it basically converts it into some internal representation, which we argue corresponds to the 350 olfactory receptors, right? And then you have another network which takes that internal representation, which are 350 olfactory receptors, and expands them back to the real space, three-dimensional space. You give it a molecule, and what you get on the exit is the same molecule. So you train the network actually to replicate the molecule itself. And how is it useful? It looks like highly redundant, right? But the most important feature is that the middle layer, which is the olfactory receptor layer, where you have the latent features that code that molecule, it's severely compressed. So the amount of information in that layer is very small. So the idea is that can you put can you compress the molecule to such a degree that basically extract the most important features in that molecule, and the molecule is still replicatable on the exit,
Right.
on the output of the network.
Sure.
Right. This is a common technique. And why is it?
It's like encoding.
It's an encoding and then decoding.
Right.
But then in the middle there is a bottleneck. There's a tiny hole through which all the information about that molecule can come in. And if you are successful in replicating the molecule using this minimalist information in the middle, then you have this very compact and efficient representation of the overall olfactory space, right? So why is it useful? And this is useful because in the visual system, when there wasn't enough classified data, so this basically relies on the unlimited data set that we have, that I described, of structures of molecules. It doesn't require any olfactory percepts, right? You don't need to know how the molecules smell to train this type of autoencoder. And therefore we can rely on the millions of molecules that we have structure for, to basically to pre-train this network for a more challenging task, which is understanding the olfactory percept. So it's basically a pre-training step. But it's also interesting, it's fascinating to know that you can compress this enormous molecular space and put it through the small hole in the middle. We estimate that you basically need about 40 receptors actually to reliably replicate the molecule with very high precision.
The end goal, though, as you said, you want to be able to classify molecules according to their smell, so you can go through this process with molecules whose smell you can describe using this semantic database. And then you can present molecules that have not been captured by the semantic database and estimate where within the smelliverse they exist.
Right.
Well, that's fascinating.
So this is like the autoencoder is the first step. Perhaps I'm a little bit too technical, but was describing all the steps. But this is basically something that we're really proud of because it works, right? So we put the molecules, we like shred them and put them through, compress them into a tiny hole in the middle of latent representation, and then we expand it and we get the three-dimensional shape perfectly, right? So that is great. But then the second step is actually to use that information, that latent information, to understand how molecules smell. So what you do is you take the part which encodes the molecule, which shrinks them to the small to a small hole, and add a classifier, another deep network, which is a classifier that will then convert that latent representation into the percept, right? So that works like fairly well, but it's not really perfect. I think there's a lot of actually room for improvement. So, we, for example, can predict pleasantness of molecule with like correlation of about 0.7. So we're about like 30% away from the perfect predictor.
Just on pleasantness.
Right.
But presumably then you have all of these words that are being used in your data will eventually be able to present
So we need
a molecule.
to go to about like 0.9 to be able to predict individual words.
Like citrus, or I was looking at some of your data. What was the one for decaying?
Warm,
cadaverous,
dusty, cadaverous. There are words like that. The ultimate goal is obviously to basically put the whole 10 million molecule database and to predict individual words for every molecule. That would be basically what we're trying to deal with. So we estimate that our predictive score has to be about 0.9 for that to be possible. So at this point we have about 0.7, but we didn't really exhaust all the possibilities for us to improve this classifier. And the autoencoder is one of the most important steps, I think, in terms of it's like one of those bells and whistles that is very important, because we're dealing with very limited data set of about 4,000 molecules, which we know how they smell. And I think for image classification, you need millions of images to build a predictive network. So we are at the point, at the very early stage, when we don't have enough data to be really predictive.
Yeah. What are some of the applications if you reach a point where you have very high accuracy probability and then you start feeding molecules beyond the 4,000 through the network? So you're classifying the smells. How do you use that? I'm sure you've thought about applications, how this can be used.
I mean, it will give us understanding of the sense of smell, right? So we will have a clear picture of what are the features which are extracted from the molecules, and that has like fundamental significance for humankind because we don't know how one our senses work.
Right.
Well, we know some of it. We know some molecular mechanisms, but we don't really understand on the same level as Helmholtz and Young understood in like early in the 19th century.
Yeah.
So we're basically like 200 years. We're delayed by 200 years compared to the visual system, right?
Yeah.
So which is ridiculous, right? So it's 21st century, we don't understand how our senses work. So that's not an application. I think that's basically one of the fundamental problems that people face, although they're probably not aware about it.
Yeah.
Right. But in terms of applications, of course, you will be able to generate molecules with given smell, which will revolutionize perfume industry. Right?
Sure. I would assume there's a correlation between the pleasantness or what's the opposite in your unpleasantness of a smell with its health effects, for example.
There's another aspect, which is the basically predicting not necessarily olfactory percepts, but for example, the toxic effect, the toxicity of different, that's what you're alluding to.
That's what I'm trying to say.
Well, there are also like data sets for that. We could use, the same network to predict toxicity, for example, for how allergenic given compounds are. And that would be also important. Obviously, it's an important application.
The receptors, when a molecule binds to the receptor, is it physically fitting? Is it the shape of the molecule that allows it to bind?
Well, that's another fascinating question that nobody knows the answer to. So there are two dominating theories, and one of them is steric theory, and another one is vibration theory. So I think steric theory is mostly accepted by like most of the community, but so the steric theory, the idea is exactly like what you said, that you have like a key and there is a keyhole, right? Molecule is the key and the receptor is the keyhole. If they fit, then the receptor is activated, right? Yeah. So it's basically like shape matching, right? I think if I were to bet, I would give that my like preference, that explanation. There's an alternative theory, which is a vibration theory, is that molecules have vibration spectrum, like basically like bending of chemical bonds, and that vibration spectrum is somehow recognized by olfactory receptor, not specifically shape. Of course, the vibration spectrum depends on shape. Yeah. But like molecules with different shapes in the same vibration spectrum could be sensed as similar molecules, right? So that's the hypothesis. Yeah. So there is some evidence for vibration theory, but it's questioned by most of the scientists. So that is one of the things which is not really understood.
And when you have the semantic database, any particular molecule, there's a range of words that people apply to that molecule. Is it the case that there are multiple receptors activated by one molecule, that maybe there are some receptors where one part of the molecule fits and then other receptors where the other part of the molecule fits, so that you get multiple...
Right. Yeah. That is what is called a combinatorial code. So the understanding is that like the same molecule will activate a large subset of receptors, perhaps somewhere from 10% to 50%. It depends also on concentration, like higher concentrations will activate larger number of receptors, like generally. And then the pattern of activations of those 350 receptors will convey the odor identity and concentration. Right. So unfortunately, I cannot be more specific because it's basically unknown. Yeah. What is the logic of that, which is not really sparse, right? So you have maybe from 10 to 50% receptors activated. Another interesting question is that like the same molecule at different concentrations is perceived as similarly, right? And what is similar between those patterns for different concentrations that makes the molecule appear as a single percept, right? Like, for example, when you try to find a coffee shop, right, you follow the gradient of coffee, of smell of coffee, right? And the smell of coffee will be similar, both like 100 meters from the coffee shop and like 10 meters from the coffee shop.
Yeah, that's fascinating, yeah.
Although the pattern of activity of those receptors will be quite different.
Yeah.
There will be more receptors activated in the latter case.
Yeah, yeah.
So what is it, how is it that we interpret it as the same thing, as the same smell of coffee, right?
Yeah.
There are some speculations about theories about how this happens.
Yeah. So going back to where you are in the research so far on the classification end, you said you're 70%?
Well, our score is about 0.7, which is like this correlation coefficient that describes how well we can like describe pleasantness of a molecule.
Yeah, just unpleasantness. Yeah. To get to the individual semantic labels, is it a matter of you need a larger data set, or it's a matter of refining the
model, the network?
Yeah.
Well, I think both. Like obviously we can improve our models, and we have some ideas of how to do it. But I think ultimately both are necessary. So I think we have some tricks, in our sleeve that we are prepared to use in the next like a few months. But also we have to have probably more data, because like 4,000 molecules is a little bit difficult.
Sure.
But only our experience will tell. Of course, like it's much easier to introduce bells and whistles into deep network. Yeah. It's much more fun than dealing with collecting experimental data, right? So I anticipate that we can basically improve our networks to as much as possible.
Are there sensors that can identify molecules now in the air? I mean, I was in Iraq, for example, and they gave us these stickers that you put on the outside of your vehicle that was supposed to detect chemical weapons. So
I think like mass spectroscopy would be, I mean, it's very expensive and heavy equipment, but it can do it. It's the one which is used in airports to detect.
Used in airports for
detecting like explosives when they swab you. But again, like the thing is that the mass of the like compound. I mean, the smells that we're exposed to, like smell of coffee, would include hundreds of components, or like a smell of lemon, like apple.
Components meaning molecules.
Individual molecules. That's another interesting and fascinating question of how they're combined. Like if you use mass spectrometer to identify components, you only know their mass. So you have to basically then resolve and understand what the individual molecules are. At this point, artificial noses are not really a viable alternative to basically biological system. My understanding is that the engineering systems are not very well suited for this purpose at this point, so they cannot really exceed human nose.
Yeah. That's fascinating. So what's next in your research?
So basically, in the next few months, we're gonna like add bells and whistles, like more bells and whistles, like explore other deep learning methods that we can learn. That's why I'm at this conference, right, to learn more. Then another interesting question is basically how to introduce mixtures, because real smells are mixtures. So that would be also a fascinating thing to add to the architecture of the network.
Wow, that's fascinating.
That's it for this week's podcast. I want to thank Alexei for his time and hope everyone listening has a renewed appreciation for their olfactory receptors and the volatile molecules that excite them. If you want to go into greater depth about the things we talked about today, you can find a transcript of this show in the program notes. I encourage you to download and read because I find a transcript captures and clarifies details that the ear misses. You can help us by rating or reviewing the podcast on whatever platform you use to listen. It helps increase our visibility and reach more listeners. The singularity may not be near, but AI is about to change your world, so pay attention.
No, I can't get close till I buy my time.